welcome you all to our very first lesson of cell biology. That is lesson 1. And this is our very first class with the Form 5s of 2026 as of February. Okay. Now, of course, this is the very first topic as far as the curriculum is concerned. Ever since 1987, the topic that is always introduced first is the biology of a cell. Because without a cell, we can't have any other biology proceeding. Of course, you know, biology is the study of life. So without the control of the cell, we can't have anything else to proceed. That's why it's the very first topic that is concentrated on. Now, as far as the curriculum is concerned, cell biology was broadened. They try to put in more topics. So nowadays, may I call it a branch of biology, cell biology, with some nice topics that we are going to talk about and also have an introduction. Hope we are together. Okay. Now, you very well know, whoever is here, that I always use the very first minute because there are people in each and every free class we have. We always get new people. So those new people always ask themselves, who is really talking to us and what is Science Scholars Academy? So I always spare a minute. So sorry to those who always listen to this, but you have no choice. Okay. Now, very fast, 45 seconds, just about us and about me. Then we have the lesson targets. We look at the syllabus book, we begin. And I'm going to tell you how biology lessons happen at SSA. It's a bit different from the math, physics, and chemistry lessons. You need to be very sober. You need to have the book already open. You should have the pen ready because a lot is said than it is actually written. Someone is sending me requests. Please stop sending those requests because I'm not going to accept them. Okay. Now, very fast, I told you that you can simply call me Dr. Paul, or students just call me Doctor. Yeah. I told you I'm just passionate about science and teaching young science scholars. That is all. So I'm just a tutor of science and math at Science Scholars Academy. Okay. And it is based in the country Uganda. So our vision is simple, is creating competent science scholars in Uganda. Of course, that's where we are based. And hope you know the meaning of competence. Okay? Now, the services we provide, it's basically homeschooling program where you study from home and simply go to school to do papers, but we assess you. So the papers I mean, it's mocks and UNEB, provided you have the determination. Homeschooling is not for everyone because it's hard to study when you have full authority over your phone and which app to open. And then we have the holiday coaching program, yeah, that usually goes for a month for first term and second term, then for 2 months for the third term holiday. Hope you get. And I told you that one usually goes for 100,000 for the whole combination plus ICT and GP. So you find out that you're studying for 5,000 per subject per week, something that is really fair for a student that is ordinary. Then the other one are the one-on-one consultation sessions, advice and counseling. Those ones are free of charge. No one will ever ask you for money for that. Yeah, because you have personal problems. So you always create time and talk to you. I sent you one of the consultation numbers. I'll send the counseling numbers of SSA. But counseling in line with academics, not any other. That is okay. So I told you the services are very clear. You can actually read through. For those who are new, you just read through. Everything is clear. But they are simply online video lessons or classes for strictly biology, chemistry, math, and physics. Live via Zoom, like this live class, it's Zoom. And when we record, we store everything on our app. And we make some of the video lessons accessible on Google Drive, which can be free or private. So the products are our summarized notes, very good notes. For example, the notes we are going to use today. Why I always want to give you this introduction. I don't want people to ask me for notes after. You're supposed to take screenshots in this very class, but some of the notes will always be provided. Like this is our senior biology book 1. Very organized and you'll never find it anywhere. That is what we are going to use today. and it is purely in line with the curriculum. And we always use it alongside the syllabus book, having the different competences and the different content or the course content. Hope we are together. So still, that's why among our products, we have those notes. They are still under review, trying to add in more standard scenarios and their possible responses so that we can officially give them to the public. Hope that is okay. So, we told you we basically concentrate on A-level scholars, Senior 5 and Senior 6, those that do science. So we cater for all science subjects. Subsidiaries are also catered for, and I told you we always facilitate them earlier. Later on, Yeah, earlier is always very brief, but later on we try to make everything so strong. So whatever we record, whatever we've been recording since 2016, we have tried to pull out the best videos ever, and we've kept them for years. So you can access them anytime of there, anywhere you want, whether at home, whether at school. The recorded classes are always there, and they are always fine-tuned to meet the new curriculum expectations or outcomes. So if you really want to get in touch, that is our email, that is our contact. Those who would want to look at or have a look at more sample free classes, you just check on our YouTube channel. Hope that is okay. Now, the timetable is very clear that we are supposed to be having cell biology getting introduced tonight, and it's what we are on. So let me hope whoever is here is on for the introduction to cell biology. Hope I'm right. Okay. Now, before I even give you the lesson targets, I'm going to first brief you on what really the branch, the one that I call the branch, which topics does it really have? But to make sure that I have it in line with the syllabus, for me I called it a topic and decided to call these other ones branches. But the best thing would be the branch and then the others topics. But whatsoever, just have it in mind, it's a very big topic having other topics within it. I don't want to call them subtopics because they are very big. Are we together? So, cell biology, very fast. How biology lessons are handled is a bit different from math. Math, we did a lot of writing. Yes, a lot of writing. But trust me, with biology, it's more of saying. You need to listen to these lessons again for you to take note and learn more biology or to make your biology better. This topic usually turns out to be hard for some students because it has words from other topics, but we shall try to say those words slowly and also say something about them briefly. So those small things we shall be saying about those words will need your writing and extreme and maximum attention for you to really get the meaning. So cell biology, it's basically having 3 of them. So I don't want you to hear cell biology and you think it's maybe something else. It is that. It is the cell biology, and it's having divisions in this order according to the syllabus book. So according to the syllabus book, the very first thing should be chemicals of life. The second thing should be microscopy. Then the third should be cytology. Then the last one should be histology. Yeah. So when you hear cell biology, don't think it's just one topic. There are really 4 topics within. So for me, I prefer this coverage. I prefer covering cytology first. That is what I prefer because that has been the norm for some good time. It's always good for someone to first know about the cell, the organelles within, their functions, and so on. Then someone can maybe go to microscopy. Then maybe someone can go for histology then, because there is a way they are connected in that order. And chemicals of life does not relate so much to all these. So for it can be covered. You can even cover it as you're covering cytology. Very possible. You get, because it doesn't have, it doesn't need much from these other three. So for it, you can cover it anytime. Actually, even concerning microscopy, I would even prefer, I'm going to have 2 or 3 for microscopy, 2 or 3, even histology, 2 or 3. So that 2 or 3 really depends on how the students you're handling perceive the cytology, how they get the cytology, so that you be like, this is the kind of students I'm dealing with. Meaning they will understand histology more than microscopy. So let me begin with this. Or they will understand microscopy more than histology. Let me begin with this. So that's what I also do. I'll first see how you're going to perceive cytology. Then I see whether I should go to histology or microscopy. Of course, depending on the content under each. But chemicals of life, it can be any time. So in the future, as we introduce chemicals of life, because we already have something on them, that's when I'll tell you the divisions that we cover: lipids, we do something on proteins, nucleic acids, enzymes, and so on, and etc. So that means that today we are here for strictly for cytology and cell biology. So your question should be, what is really the difference between the two? I thought it's the same. Cell biology is really biology of the cell. But then cytology really looks at the structure of the cell. Yes, and then maybe the possible functions or function whatsoever. And that's what we are going to introduce today. So, what are those things that are under the topic? Now you know that we are going to talk about cytology. Is it okay? Now, the cytology is supposed to be introduced. Then after introducing it, we need to know the divisions and how to handle them. Yeah. So basically, for cytology, we look at 2 things. We look at, shall see, we look at the prokaryotes, we look at the eukaryotes. And then I will have to tell you, we shall see that in our notes, that these ones are the types of cells. To do that for cytology, we strictly look at the structure of a cell, that is the organelles, or what a cell is really composed of, and the function of each component. So the types of cells we are going to look at. So after looking at them, of course, we look at, or after we compare. And then someone may be like, how do we really look at them? Of course, we also look at their divisions. We look at their divisions, we look at structure, etc. I told you that I'm going to be saying a lot. Then I'm actually writing to save time. Because if I write as I speak, the biology is too much. We may sleep here. So for prokaryotes, we shall see the different divisions of bacteria that are here. Then with the eukaryotes, the divisions, of course, we have the plant cells and animal cells. Now, like for example, I've not written that, but you've heard that eukaryotes, plant and animal cells. Hope people read this as you. You can check Google. Go to Google and put in pronunciation of eukaryotes. You hear the word. I don't want you to be very weird students who read this alone and this alone. Okay? Some people say eukaryotes, but I want you to be very good students and say eukaryotes. It's strictly eukaryotes. Like you say Europe. You get Europe. The whole of this is read as what? Europe. So even this one is eukaryotes. Are we together? Okay, now concerning their structure, that means we look at the structure of a bacterium cell under prokaryotes. We look at the structure of the plant cell. We look at the structure of the animal cell under eukaryotes. Then after we compare, look at function and other things. Structure, function, etc. But structure and divisions are so important. Then after we compare, let me hope people are aware. Those are the words we talk about in question approach, but you should be aware that by comparison, two things are required. They always want to look at the similarities and then also the differences. So if they tell you to compare the structure, you should be knowing that. Then the differences. Someone may be like, but I thought that is compare and contrast. It is the same thing, whether compare and contrast, the same thing. So hope you now realize what we are really going to look at under cytology. Of course, by the time those other things come in, we have introduced the topic. So the introduction is also very paramount. But I want you to be ready to listen to a lot of content. Yes, biology, we always cover it in different ways, but the most common ways is us reading the notes together, especially for topics where the wording is too much and a lot of explanation is not needed. We always just do what? Read the notes as we explain. Then for topics where the wording is less and there is more of understanding, like coordination, we really first say the biology together and then go again and read the notes. Is it okay? Then for topics that really too detailed, we always read textbooks. We can say that for this lesson, we are going to read BS. We get the biological science, we read it together as I teach how to summarize. I did all that with the Form 6 topics. You know, some of you are attending this, but you're not aware that the Form 6 topics actually got done. So whoever is attending to this, be aware that Form 6 topics were done. So we are going to just cover Form 5 topics, like maybe up to first term holiday or second term holiday, and we complete. Then by the time you come back for third term holiday, you just straight away access the topics of senior 6 and watch. For maybe like which ones are those? When you look at your syllabus book, biology topics are only 9. I don't know if you're aware. I told you that in the meeting, and I always told you that. The second topic is nutrition. Anyway, I told you all that. I don't really expect people to be having hard time with that. The second is nutrition. The third is transport in humans. We have respiration as the fourth. We have the fifth as homeostasis. We have the sixth as coordination. We have the seventh as inheritance and evolution. We have the eighth as growth in plants and development in insects. And we have the ninth as ecology. The free drive link I gave you has somewhere the SSA library, and we have syllabus books folder where those topics are. Hope people are aware. So by now, I believe people should be knowing that Senior 5 has 5 topics, Senior 6 has 4 topics. And I want to assure you that the 4 topics, first we covered 80% of them, Senior 6. Yeah? So, since you people are my friends, let me just try to show you something small and let you know that the work is just there ready for you. It's you guys who are delaying to actually have access to it. Okay? So just something small here. I told you that we have a Google Drive where our work is stored, but we always try to give you a few topics that those we really decide that let students have this for free. So you can see that biology is somewhere here. Do you see it? That one. So when I click, you can see Senior 6 Term 1 work, Term 2 and Term 3. Term 1 work, you can see coordination in animals. You can see the classes. They are Lesson 1, Lesson 2 and 3. Remember, it is new curriculum content. So you actually have those topics ready. So don't wait for your teacher to teach coordination in class. Get an idea. Go to class, sit back, listen to what you already know. There is no way you can now be beaten in the subject, provided you now read again like 3 times. It's about repetition. But depending on our level of memory retention, there are people who need to read once, there are those who need to read 2 times, 3 times, and so on. But I always told you that repetition is key. If you have been listening to me since day one, time to work, you can see the work is there. Inheritance and evolution, nucleic acids, lessons are there. Don't wait for your teachers to teach. Learn it first, go back, learn it for the second time. Come back and watch again, now consult again. And you will not fail. The problem is that all those tasks, you can see even cell division, everything is there. That is term 2 work. Even term 3 work, we have there ecology. Now, just imagine we have now started our journey on senior 5 work. And I've told you there are those ones we already have, though they are too much into the old curriculum, but they also work. But trust me, by the second term, every topic will have been repeated. In senior 5, these of Form 6 were repeated and are in line with the curriculum. So you realize you have a lot of work to cover ahead of you. So don't be these students who spend a lot of time on TikTok and on unnecessary things. Hope we are together. Today I'm leaving you with research work. Just look in for the 2 years. They really run so fast. Look in for the 2 years. Get the dream course. Get the points that can at least get you a course on private, at least. But struggle from day one to the last day. So that even God realizes that, yeah, my child tried. But the moment you become a mediocre along the way, you're really losing it all. Just stay with me for the 2 years. We are going to move together. And trust me, we shall make it. Yeah, because I made it and I know what is really supposed to be done. So you'll be learning from my content and from my mistakes, even when I passed. Okay? Now, back. Hope my support assistants are on trying to collect concerns from the chat that we shall address later on. So people have gotten an introduction of the topic and you know the order that we are really going to follow. So, the points of the syllabus book, that is the book. So you can see section 1, competence. I told you these competences, we shall use them in revision. That's why I want to cover those topics of senior 5 with the people who are going to stay for homeschooling. So that by the time you come for senior 5, term 1 holiday, we are done with more than 50%. So we shall just revise. And you realize that as we revise, these are the words we shall concentrate on. That if at all we are to do a question of cell biology and it is under the very first section, we shall need such competence. That it should be in line with analyzing properties and functions of chemical compounds, blah, 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 and so on and so forth. The way it is. But now let me read that of the topic we are covering today. So I've told you that the topic we are covering today is under section 3. Isn't it? And what are we really supposed to do to analyze ultrastructure? By ultrastructure, they need you to look at how exactly the organelles look like and how maybe the structures even relate to their functions. You get? And later on in the end, we need to compare what they call the distinguishing. Is it okay? And then even what are the things that the syllabus really wants us to do? It needs us to study and compare images. Which images we are going to look at. I want you all after this lesson to download the app called Pinterest. Pinterest. This is where I get my images in most of the cases. Pinterest app. I want my support assistants to go and look for this app, look at how it looks like, encourage people to download it. Now when you sign in, you can ask it for any image that you want. And you can keep editing your prompt. The prompt is what you put in. So you can ask it for image of ultrastructure of animal cell, image of ultrastructure of plant cell, bacterium cell, things like that. It will provide everything. It's having good images, very good images from very good textbooks. So provided for you realize that what it has provided is in line with what you need, you just know it's perfect. And you can save those images to your device and keep them. You can have your own WhatsApp group where you put your work and you be organized. For me, I had my WhatsApp group for the 2 years. After passing, I just deleted the group. Then I rested for a year and then came back to educate the young generation. So that app is very important. So the syllabus has all what we have to cover, but we also have a way to cover them. We may not follow the order, but we follow a way that will make you understand. But in the due course, everything will be what? Covered. You get? So for today, we are really interested in the prokaryotes. Have that in mind. Of course, the ultrastructure, we are going to look at functions, then also the divisions. These are words that the people of old curriculum never looked at. The eubacteria and archibacteria. Try to ask any of the people who are in vacation right now of senior 6. They have never heard of that. This is something new in biology. Because as you know, classification is like technology. It keeps on changing and changing. They keep on reclassifying organisms. So we are going to look at how and who broke down the kingdom Monera into two kingdoms, the eubacteria and archibacteria. And there is very good biology there. And I think I've gotten an idea. I'm going to do one kingdom and then I'll give you the other kingdom as research. And then I'll give you a task of comparing the two. I think that's what I'm going to do. And then we shall be done with prokaryotes. So that our next class we have eukaryotes. And then maybe we shall be 70% or 80% into the topic. Then we see what we can really handle next. But let me hope people are reading as far as the instructions we always give you are concerned. Okay? Now, hope you are all aware of what we are really going to do. So we are now going to read our notes together. And the moment we find any statement that doesn't make sense, we do the needful. Raise up your hand, we explain. So this is strictly cytology. Yeah, cytology. Okay, now that is our biology book 1. And I'll go to the very first page I extracted. Okay, so you can see the very first topic in the syllabus book, cell biology, and section 3 is cytology. But what does it really have to look at? Why I'm explaining these notes is that after this lesson, I'm supposed to provide them, but I'll provide them tomorrow. So those who are on, use this chance to be taking screenshots. I'll also allow you to take the screenshots. Then maybe by tomorrow, by the time people are reviewing the recorded work, they can, okay, the link will be ready to download them. Is it okay? Because now this is a full book, but I only want to provide one topic. So I need to first extract and edit. Okay? Now, what does it really have to look at is the ultrastructure of the prokaryotic cell, bacterial cell. The bacterial cell is now a form of the prokaryotic cells, and the eukaryotic cells, or the eukaryotes, prokaryotic cells, eukaryotic cells. Okay? These ones I told it's the plant and animal cells. Together, don't write in my screen. Don't write in my screen, please. Every time you write in the screen, I'm supposed to close it and reopen it, and that is time wasted. So please at least bear with our time if you can't bear with us. So I'll come back later to this structure. Now, you seeing this diagram? I got it from Pinterest, but it is very good, and imagine it's part of my book. So that means Pinterest is very good, and it will give even better ones. Okay, I'll come back to that structure later on. Let's first read through. Yeah, let's first read through. So, you all know whatever we are handling right now is what? Cytology. So what is this cytology? Now, content has started. Listen, have the book and pen ready. The moment I leave you, you're gone. So they are saying the study of cell structure and function of functions is what they call cytology. That is the definition. You all know that logy means study. So cyto is strictly for the cell, and two things, that is the structure and the function. What are they saying? The cells are so diverse. I've told you why we are reading the notes. I'm supposed to provide them. So here it's like you're reading. So those people who always read ahead, what we are doing is also reading ahead, but I'm reading with you as I'm explaining. So you're supposed to also read the same notes after, so that as you read, you recall the words I was saying. But you're supposed to do that immediately. If you're too late within 2 or 3 days, the moment you do it in a week's time, you won't be recalling the words I said. And you cannot be the same biology student as that one who read them immediately that night when the lesson was held. Hope we are together. So they are saying the cells are so diverse in both structure and function. By diverse, they mean variety. That you realize cells don't have the same structure, and they have different organelles with different functions. That's what they mean by diversity, variety. But they are saying that basically, normally they are what? Spherical. They are saying that though some of them may show variations, and these variations are because they want to suit their different functions. You find out that cells don't have the same function. So to suit the different function, that means we may not need the same structure. Hope you understand. You're living in Kampala, I'm living in the village. You like it or not, our cost of living cannot be the same. That means we may need different jobs, even when we are both in the same country. So the spherical shape is the basic shape of cells. But because some cells take on other functions from maybe those we expect them to have, they always show some variations in structure and function, and we shall see which cells as we progress. One of the cells we looked at is the nerve cell under coordination, which is already covered. Okay, so what is the definition of a cell? So the cell is defined as the smallest, first and foremost, that is a keyword, basic or fundamental keyword, structure and function. Why does structure move with function? Because the moment you lose structure, the moment anything loses structure, its function is lost. For example, if you know a cell, and you all know that a cell has what we call the mitochondrion, which is singular. Which process happens in the mitochondrion? For the doctors who are here, you people want to become doctors. Okay? You people, don't you know what happens in the mitochondrion or in the mitochondria? Yeah, Josh Best has tried, has said respiration, and we are going to see that. Thank you so much. So that means if, remember the mitochondrion is an organelle. By organelle, we mean what the cell has. They are the things we call organelles. So those organelles have a specific structure and they have a specific function. So realize if at all respiration is taking place in the mitochondrion, if at all it gets damaged, so it may, its structure may be damaged. So the function, which is respiration, may not proceed. So in most of the cases, structure moves with function. So the cell is the smallest basic structure and functional unit of an organism. There is a topic they removed from your curriculum. I told you this from the meeting, but you may need to read the topics afterwards. Yeah, but they are what they call levels of organization. That if at all you have a cell, I think you even look at this at O level, the group of cells that have the same function or that serve whatever similar characteristics and so on, when they are together as a group, they become a tissue. The levels of organization. And then when also these tissues are a group and serve a similar function, they become what we call the organ. And then when also these different organs are trying to come together as a group to perform one sole function, they become what they call the organ what? The organ system. Is it okay? And then the organ systems together to make sure that the organism survives. So all together they give us an organism. But when you reach medical school, we realize that we have some things before the cells. But that is medical school. Is it okay? Now, you can realize that the cell is the first level of organization. So you like it or not, is the smallest of an organism. So an organism has organ systems, an organism has organs, an organism has tissues, an organism has cells, and the cells are the smallest. For example, you the person, you are the organism. You have organ systems, for example, your digestive system, your reproductive system, your excretory system. You have organs, you have the heart, you have the skin. Some people think the skin is not an organ. You have the liver, you have so many things. You have tissues, blood. I don't know whether people are aware that blood is a tissue. Then you have cells. Have you heard of liver cells? Do you know the specific name given to liver cells? All of those things are taught in homeostasis, where we talk about the liver in detail. But you meet all those. So you realize I've said a lot. So you need to be taking note very fast, or you promise yourself to review the class just to take note of that biology. In the future, I will need it. So on top of our very good summarized notes, you also need a summary, but just to supplement whatever I've said. So we have what we call the modern cell theory. The modern cell theory, or just simply the cell theory, why I'm calling it the modern, it's the current one. But as I've said, biology, most of the things, especially that are to do with classification, evolution, they always change with time. So this, the current one or the modern one has 5 components. So they usually tell you to state the modern cell theory. You state the 5 components, and we are going to talk about them. So someone may be like, you're saying this thing of what is it? So the modern cell theory simply talks about the facts about cells. Yeah. So don't overthink. So it's a theory, don't think it's one. Yeah. It's a theory, yes, it is one, but it has so many facts. And those facts are about cells, and they are modernly 5. Hope we are together. So what are those, and what do they say? So when they say state the modern cell theory, you state the 5 facts. Okay? Now, what are they saying? They are saying that all living organisms, when they say living organisms, think about yourself to make things very easy. So they are saying that all living organisms are composed of either. Why you need to fix there either? Because of this all. So it's either one cell like the bacteria. The bacteria simply, they are unicellular. They are just single cells existing on their own without finding any issues. Trust me, the modern cell theory is one of the things they really think about as they are setting for you biology. And now A-level content has started. So don't even blink. Okay? So by composition of one cell is what they call the unicellularity, or just being unicellular. Then they can be 2 or more cells, or what we may just group as more cells, simply meaning more than one. And that's what they call multicellular. One cell like this, single celled. Are we together? Then more cells, multicellular. The many-celled organisms. I think in O-level we used to call them single-celled organisms, and then the multiple or many-celled organisms, things like that. That is the very first component, or the postulate of the modern cell theory. Okay, now all new cells are derived from pre-existing ones. Note that cells just come as rain. They don't just come as magic. It's the pre-existing ones that really divide to give us other cells. I think as a person who has ever attended biology at ordinary level, you've heard of cells dividing to give you more cells. You've heard of things like binary fission. Okay, you've heard of cells dividing to give you more cells. So that means it is a cell that is already existing that will give rise to the new cells. Hope that is very clear. And you state them the way they are. What next? You people, you've heard of what they call heredity. Heredity is inheriting some traits. You've heard of DNA tests for people who are not sure whether the children are theirs. You've heard of things like the child resembled the mother. The boy took the traits of the father. You've heard of things like that. Why it really happens? Because, okay, the mere fact that they tell a certain child that they took on traits or any body part, it can be the fingers, it can be the face, it could be the nose, it could be the legs. They'd be like, these legs are for the father, this nose is for the mother, this forehead is for the dad, things like that. So that means there is something that really helps us to transfer those traits and so on from the parent to the child. Are we together? And that's what they call inheritance. It's like the child has inherited something from the parent. So who is really responsible for this? They are cells. Remember, they are the basic stuff in the life of an organism. But what exactly helps the cell to do that? To help us transfer those traits and so on. So we are saying that these traits have what they call hereditary what? Material. You get? That hereditary material is genetic. Sometimes they call it hereditary material or genetic material. That's why they call it that's why they call it a DNA test, or they call them DNA tests. That's why they tell you that that child is yours. Don't even waste time to go for DNA test, because you can look at the forehead. That forehead is yours. So that means DNA, heredity, they are just connected. So you see this? They are saying that, what is the third postulate? They are saying that, or the component, cells contain hereditary material of the organism, which is passed on from parents to daughters, parent cells to daughter cells. All people are aware that when a cell divides to give you two cells, the cell that has divided is what they call the parent cell. The other two being produced are the ones they call daughter cells. Do you know that? Okay. Now they are saying that material, that hereditary material, is genetic, or you can call it genetic material. And it is called DNA, or it can be RNA. Now, this NA, you realize it is common on both. That NA stands for nucleic acid, N for nucleic, A for acid. So this topic is called nucleic acids, and if you were very keen, I showed you that topic under Senior 6, and it was covered. That's why I told you that what really makes this topic very hard, it carries a lot of information from other topics and they come here. But we always say something small about them and also say them slowly so that you don't really burn out as early as this. Then what these mean, they are talked about in that very class. And I don't want to say a lot, but R stands for ribose and D stands for deoxyribose. If you feel like you really want to learn, or if you confirm that your school is going to do nucleic acids in term 1, you send me a message and tell me, Doctor, give me access to nucleic acids. My teacher is covering them in term 1. You also watch. Before school, you go and you're the one with content. But just do one thing: when you go to school and you know the work, don't brag. When you brag, your fellow students will put you on pressure. They will be waiting for you to just fail. But even if you don't fail, but you don't be the best, they will still stress you. So the best thing is to know your work and keep quiet, be low key. So that you fail, you're low key. You pass, low key. But the moment you reach school and you show everyone that you know the work, those who don't know the work will wait for your results. And when you don't pass, they will start saying words. They'll be like, that girl was saying a lot of things at the beginning of the term, but over what has happened, she got 60, but the other people got 90s, but for them they were quiet and non-talking. So the best thing is to stay silent. When results are good, be silent. Results are bad, stay silent. Even when teachers are teaching and the teacher makes a mistake, don't correct them. Even when they say something contrary from what you understood, first wait, go to them in person. Don't raise up your hand in class thinking you can embarrass a teacher. But I will advise you, make sure before you go to school, I put for you a session just to advise you. I tell you all mistake students have been telling me since 2016 about how they misbehave academically at school and end up. So learn from people's mistakes, not yours. But I'll tell you that I really want to discuss content. So that genetic material is DNA, but DNA has a relative RNA. D and R, what they mean, I said them. If you really want to know more, I've told you the topic. It's Form 6 topic. Is it okay? They are saying that all metabolic reactions take place within cells. Metabolic reactions are divided into two. We have the catabolic and anabolic. Catabolic is breakdown. For example, during respiration, you eat food, you sleep, respiration takes place, it releases energy. You wake up when you are energetic. Are we together? You eat food, you become tired, you want to sleep. Digestion takes place, respiration takes place. The food you took in was complex, but it has broken down into simpler products and they have been metabolized. So from simple to complex, that is catabolism. Then anabolism, it is the reverse of catabolism, but they're also supposed to be talked about in details and further topics, as we shall see. So all those kinds of reactions take place within the cell. You get, look at respiration in the mitochondria within a cell, it happens there. Look at plant cells, there's what they call photosynthesis. Photosynthesis, you are photosynthesizing. It is biosynthesis. Biosynthesis is anabolism. Because you're from simple to complex. What are the simple? Carbon dioxide and water. What is the complex? Starch. That is anabolism, the reverse of catabolism. Are you listening? But where does it take place? In the chloroplast. Where is it? Inside a cell. So all that happens where? Within. What is the third? The mere fact that a cell has the ability to take on metabolic reaction, that means it can survive independently. Yes. That's why they're telling you capable of surviving. How? Independently. The smallest unit of an organism, we know that. And it is capable of surviving independently. Simply because it can do the metabolism on its own. So does it really need anyone? No. It's like saying an independent woman doesn't need a relationship to really survive. Okay? Now the note. What is this note saying? Cells perform different functions because each cell can control, to some extent, which molecules allow in and which to exclude. Do you understand that? That if the mitochondria have respiration as their role, they should only and only allow in materials they can respire. Are we together? They should only and only allow in molecules that are going to participate in respiration. And they will only and only exclude the metabolic wastes. Of course, respiration is metabolism, and metabolism usually gives off wastes. And wastes are supposed to none other than being excluded. Hope you know that. That's why they're telling you that cells perform different functions. Because right from their organelles and even the cells themselves, they have what they call the cell membrane. I think you people have heard of the cell membrane being selectively permeable. Selectively permeable. I know this is not a new word. And I remember, or you remember, that by selectively they mean that the membrane allows some materials to move in, but not others. And it allows others to move out and not others. And you see that especially in coordination under impulse transmission in the future. So after getting these notes, you should attach here something like impulse transmission, knowing you will have to understand what do I mean by the selective permeability. Hope you are together there. If at all anything is not clear, raise up your hand. Immediately, we rectify it. When I see no one raising up their hand, I believe everything is just what? Is just okay. The types of cells. So the types of cells are only two, the prokaryotes and the eukaryotes. I told you about that. Yeah, it's like telling you types of farming. So these are types of cells. Just understand karyotic. Here they are saying pro, here they are saying you. Now I want people to go to Google. You know, you people have a tendency of saying that things are hard, but things are hard only and only if you have no knowledge of how you can really bring a difference onto them. So you have all realized that the difference is on pro and you, but both of them have the word karyotic. I want you people to go to the Google or to the internet, anywhere, or to AI and ask the meaning of the word karyotic. Yeah, you can put in karyot, you can put in karyo, you can put in karyotic, ask for the meaning. Then after, you ask for the meaning of pro and the meaning of you alone. Yeah. Then we handle the prokaryotic cell and we are going to be done very fast. Trust me, we are not wasting a lot of time here. Yeah, just be ready, have your pens, books, listen to the content. Whatever is not in the books, you immediately take note. Yeah. If at all I start speaking a lot of content that is not in line with the curriculum, you tell me. I usually find one problem in biology. There is when I start talking content of medical school and you end up understanding. So it is good, but again, what you're hearing is not examinable. But you're learning because you understand. Yeah, because sometimes I forget. Because remember, we also teach junior doctors. So if we teach junior doctors, sometimes I forget that I'm now with Form 5s who haven't even joined school. Someone sent me one message having all things. Can you please rephrase the cell theory? You people, let me tell you this. You're not supposed to memorize everything now. No. Just make sure each and every statement I read, you understand. Are we together? Whatever I read, you understand. Whatever I read, you understand. It is you later on to revisit the same work and now try to memorize because you understood. So you don't need to memorize the cell theory now. No. Understand each and every component. I explain, you understand, you proceed. Afterwards, you're supposed to read. Don't be surprised when you cannot say anything after the class, yet you attended and you thought you had gotten everything. It is normal. But here you're just appreciating. Whatever we say is clear. Whatever we say is clear. Afterwards, you do the needful. Okay, have people told me I need one single message with the meaning of karyotic, any, then pro and you. Pro before you means true. But are you getting things from the internet? Then karyot. But it is quite absurd for people to be here in very big numbers and they cannot really respond to that. Just going to the internet and you ask those things, is that really hard? That is a turn off if at all I'm dealing with people who are not focused. I really want to give you a task and you respond immediately. Hope that is okay. So I'm waiting. Let me see. Seems I'm dealing with very disorganized doctors. Very bad people. I don't know where my math students I taught in the morning. They were more active. I expected doctors to be super active. So by karyotic, it's all about presence of a nucleus, just a nucleus. Then you know that U simply means true. Yeah? So those ones we shall realize that they have a true nucleus. Then pro means something that before it can mean, okay, pro means pre. Pre. You know, pre means a lot of things. You've heard of things like premature, what they pronounce as premature, something that is not yet ready. Yeah? If they say that the baby was born prematurely, that means that baby wasn't true. That baby wasn't ready. So you realize that just like the way we say that eukaryotes have a true nucleus, so the other ones don't have a true one. True nucleus, these ones have. These ones true nucleus, they don't have. Pre, pro, before, not yet there, not yet real, things like that. And for today, so that has been the introduction, as I promised. So I'm going to come in our workbook here, and I'm going to tick the introduction. The introduction is done. The introduction is done. So the next thing to cover the prokaryotic cells or the prokaryotes. And then next time, we shall be doing the eukaryotes, and then we shall end with the comparison. So prokaryotes, how do we introduce this? You already know something about them. So I'm going to first show you how, okay, have it in mind at least in whatever you're doing, or the very fact you should get from this. That's why even if before the lesson in the channel, I told you that at least come when you know the structure of a bacterium cell. So have it in mind. The very first example or the example of prokaryotic cells you study at A level that you really concentrate on are the bacterial cells, or simply bacteria, like that. Simply bacteria or bacterial cell. Singular bacterium. So that means whatever we are going to be talking about concerns the bacterial cells. So when you go to Pinterest, you can see the images very well. But again, also make your learning fun. Always go to YouTube, like I'm going to show you here. Go to YouTube and search. Tell YouTube, sorry. Okay, I want you to look here. I want you to look here. So always go to YouTube. I've said make your learning fun. If you know that you're now going to concentrate on a bacterium cell as far as prokaryotes are concerned, first learn about the structure. Have a video. Go to Pinterest. Look at the pictures. I always tell people you cannot do these things at school. The reason that's why people fail A level, people think A level is actually passed from school. You can pass A level from school, but from some schools, and those schools are very few. I don't want to mention schools here, but they are not more than 10 in Uganda. Schools where a student can really pass, even when they have not had any form of extra effort. So most of the schools and most of the students in the other schools are not aware that it's non-schools that really make the maximum points. I'm talking of maximum. It is holidays and the time of school. You're supposed to be planning for your time at school for you to get results from school. So you're supposed to use the chance when you're in your own time as a student to really perfect whatever you may need when you're now enclosed in a school system. The day you will realize that and understand that, you will be a perfect student. That's why I'm forcing you to know term one work, so that when you leave school, you now come and cover term two work. You leave school, you now come and cover term three work, like that. And the moment you come back for term three holiday, we are completing the syllabus. But if you don't copy notes, if you don't read and read and read, you will not benefit. I'm tired of advising you. Okay? Now, go to YouTube and put in ultrastructure. People are seeing my screen of a bacterium. Okay, it's already there. So when you put in bact, you can first stop there. Then you look at all the words they are bringing. I think we need a cell. So I think the first one will be better. So you click. They are going to bring you so many videos. And I've always told you that always go for that video with a lot of views. That means it is very good. Or you can watch all of them and don't go for long ones. Now this one is 6 minutes. Those are too many. This one is even 1 hour. You just know that one is not for your level. These ones are long, 8. Now, you go for these short videos. They are like TikTok videos. You watch some. In most of the cases, the best things to watch are the 3D animations. So you can even come and say 3D animation. 3D animation. Let's see if they have of a bacterial cell. Let's see if they have. So this one is 3D bacteria 3D animation. You see? Very good video. We can first watch. It's just 41 seconds. Remember, you're not going here to learn. You just want to watch something, have a look at it, so that as you read, you relate. As you read and relate, it really makes your reading just perfect. Okay? So we are going to watch this video together. So you keep quiet and listen. Just 41 seconds. So I'll click.
Imagine conducting every part of your research only on one platform. Meet Oreo.
Okay, you listen. You just watch. Have you seen how it looks like? Now, I want to first pause it. I don't know. Are you seeing that? Someone may be asking themselves, what are those things that are on? Those things are called pilli, or people simply call them pilli. Now, I want you to look at it. It's having some other longer one that is longer than all of them. There are those ones that are there on the cell like that. Those hairs, they are called pilli. But then there is a longer one that is longer than all of them. I don't know if people will see it, but let me play the video. I don't know whether people are you seeing those longer ones behind? They are those ones that are causing it to move. And they are the ones they call the flagella. Flagella is plural and singular, it's flagellum. Are you seeing it? Now this one they are trying to show us is rod-shaped. This video has been very good. I want you to I want us to watch it again. I want you to watch it again. There's something else I want to show you. So those ones are the pilli of fimbri. Fimbri. You're going to see all that. The other longer ones behind, they are the flagella. Singular flagellum. You take a screenshot and tell your friends we are watching a movie. Let them come and study. So you can see the structure is rod-shaped. Now, this is where I wanted people to see. Let me wait and see here. Now, you look at that. Right now, they cannot show us the layers, but it has layers. It's not alone. But at least you've looked at the pillar, you've looked at the flagella. Let me first, there. Now, are you seeing those things inside? Let me ask yourself, what are those? There are those things that are also inside that they are taking in. Let's first wait for, we can, oh. So those things you're seeing, the red ones, they are later on going to come together. and they are going to be somewhere in the center. The other ones are just, they can be anything. They're just inclusions. Inclusions are the things included inside the cell that are not other organelles, but we are going to look at the diagram, and trust me, you're going to get it. So those red things you're seeing, they're later going to come together, and those things you're seeing, they are the DNA. Yeah, and they are going to come together and they are going to coil together, and you're going to see their name. They are called the nucleoid. Biology has a lot of terms that you really forget, but when you over repeat them, time will reach and they are just obvious. So I'm also giving you a task to later on. Are you seeing how they are coming together? Are you seeing? Now they have disappeared. So there are so many videos. You can find time, you can watch. But please try to make your education a bit fun by going for such videos. There are so many. There are so many. There are so many. I don't know. Let me see. There are the other short videos I told you. Let's also watch them. We are doctors, we have the time. Okay. Are you seeing? Oh, these are videos. Very good videos. And I want you people to watch them, please, in your free time. But the last one we are going to watch is this one of 1 minute and 39 seconds. Yeah, I think this one is going to be very good. Let's see if
Serious
it
dictation
helps.
is driving me crazy.
Okay.
So I switched to whisper flow. Here's why I'll never go back.
Welcome to Vedifile everyone. Today's topic is bacteria cell structure. They are as unrelated to human beings as living things can be. But bacteria are essential to human life and life on planet Earth. Capsule. Some species of bacteria have a third protective covering, a capsule made up of polysaccharides. Cytoplasm. Vedifile everyone.
I don't know whether people are listening to what that lady is saying. You're supposed to listen because they are the same things you are going to find in the notes. She has said that those bacterial cells are very important, just like human cells. She has talked of their structure. I think you've seen the red lines between that have been together, the ones I told you about. She is now talking about the 3 layers. And you have realized that outside completely we have the capsule. In the middle we have the cell wall. Then lastly we have the cell membrane. Please tell Kato Emmanuel to leave the screen. I'm going to remove him from the meeting. I don't know why he's excited. Then it's going to also show you what is inside. So you watch again.
Today's topic is bacteria cell structure. They are as unrelated to human beings as living things can be. But bacteria are essential to human life and life on planet Earth. Capsule. Some species of bacteria have a...
Are you seeing the capsule completely outside, the one in white? Then do you see the cell wall next? The one in, I think, brown. Then that one in yellow, and it's called the plasma membrane. Are we together? Because you're going to look at it again in our notes. So you don't need to be lost. Okay, let me see what Dalton is saying. Yes, Dalton, but be fast because we don't have time.
Excuse me, sir, we can't see the screen.
You can't see the screen. I doubt. Isn't it his device? You tell me, you tell me the person. You tell me the name of the person. You tell me the name of the person. Sorry for that. Okay. Let me remove them. Thank you. Okay. Thank you for that. But no one has called me, and I told you for that, for such disturbances, you're supposed to give me a call. I don't know where now did you stop. Thank you, Dalton. Where did we stop? Or can you see the capsule outside, the yellow one? Sorry, the white one. Then the cell wall in the middle, the one in brown, and the plasma membrane, the one in yellow. The one that immediately encloses whatever is inside. Can we now begin the video from there? Have you looked at the 3 layers of a bacterium cell? I told you it's today's topic, so we really need to give the structure time. After all, even the competence is about analyzing structure and the functions. I don't forgive anyone who writes in the screen. They wait for the recording. I hate disorganized people. I think we can begin from there.
Third protective covering, a capsule made up of polysaccharides. Cytoplasm or protoplasm of bacterial cells is where the functions of cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water, enzymes, nutrients, wastes, and gases, and contains cell structures such as ribosomes.
Have you heard? They have talked so much about the cytoplasm, whatever it's having. They have told you it's gel-like. They have told you a lot. They even told you that the capsule has polysaccharides, but we shall modify that. Okay, they have said a lot about it, and we shall also read through the notes and you get to hear. But just look at it. It's the one that is having many things. They have said enzymes, they have said water, fluid, organelles, and so many. Now they are starting to mention the organelles that are within the cytoplasm. And they are beginning with ribosomes. So the moment the video proceeds, they are talking about ribosomes. So you gotta listen.
Chromosome and plasmids. Flagella. Flagella are and contains cell structures such as ribosomes.
I want people to look at the plasmid. I don't know. Do you see the plasmid? The one they call the plasmid? Let me play the video. You look at it because it is also very key in this lesson and it is very key in your curriculum.
A chromosome and plasmids.
That carrying that is surrounded by some things that are colored blue and red, I think. That is the plasmid. So when you look at it, it's just a ring. So when we are reading and they call plasmid a ring, I don't want you to be confused as to why they are calling it a ring. And it is a ring of DNA. And we are going to see what it really does. Very important. Okay?
Flagella. Flagella are hair-like structures that provide a means of locomotion for.
Have you looked at the flagella? The ones I told you that are the longest or the longer ones. They are the ones that are responsible for movement and locomotion. Then these smaller ones around the cell, they are called the pilli. Pilli, can say pilli, whatever. See, it's singular, it's pilus or pillars. Then fimbri, plural, singular, whatever. But the spelling is more important. Okay, we proceed.
Those bacteria that have them. Pilli. Many species of bacteria have pilli, small hair-like projections emerging from the outside cell surface. Ribosomes. Ribosomes are microscopic factories found in all cells.
Have you heard? Ribosomes are microscopic. The pilli are hair-like projections from the outer surface. And you're going to see their role. They're important for attachment, the way you see them. So they are coming from the capsule. That means even the capsule is key in attachment. And they're also responsible for sexual reproduction in bacteria because they are key in attachment. So relating them to sexual reproduction is the easiest. Okay? Proceed.
Bacteria. They translate the genetic code from the molecular language of nucleic acid, that of amino acids, building blocks of proteins. For more information, like and follow our Facebook and Instagram handle.
Hope that is okay now. Hope that is okay now. So that last language you had of the ribosome converting the molecular DNA information into the information of amino acids, that is now under nucleic acids. And we also taught it's called protein synthesis. It's there already, we have it. Is it okay? Now, you now know a lot about the structure. You now know a lot of things. Now we are going to just be going through stuff. And we are just going to be enjoying. Is it okay? Now, what are they saying? Prokaryotic cells, where the very best example, the last bones are the bacterial cells, they have the following distinguishing characteristics. We mean the characteristics that distinguish them from other types of cells. Are we together? What is the first thing? They lack double membrane bound organelles. I told you that the word organelles simply means those things that are inside the cell that have a particular structure and function. And I used an example of the mitochondrion, plural mitochondria, that they have their own structure. And you all know their function is to handle respiration. So anything inside the cell, even if it's not inside, but anything part of the cell, and it's having a particular structure and function. We shall see things like structure of the nucleus, structure of all these things. You see that their structures are distinct. When you read FA and BS, they define an organelle as a distinct part of a cell that has a specific or distinct structure and function. So they are saying that these ones, pro, I told you something premature, not yet, there are things like that. So most of the things they actually lack. And the very first thing they are known for is their nuclei lacking a double membrane. One of the things you're supposed to really appreciate is that they don't have a true nucleus. Why is this nucleus not called true? Because a normal nucleus is supposed to have a double membrane. To tell you this, or to give you some lugam or some secret, this is how the nucleus looks like. Give you some lugam. Because most of you are not sure, let me say about it so that people can understand. That's how the nucleus looks like. Otherwise, most people know that it looks like this, but this is not right. That's why they call it microscopic. That for you, when you really analyze it or examine it under a microscope, you realize it's having those what? Nuclear pores. Yeah, where entry and exit of material is regulated. Then you can also look at its membrane. It's double membraned. The double membrane is bound by 2 layers. Even the mitochondria, most of you know it. When you go to Pinterest and ask for the images of the mitochondria, they will bring it. Even if you go and check now, it will be like this. Most of you are aware it looks like this. If at all your biology teacher tried to show you this in O level, it looks like that. This outer one is the first membrane. This one is the second one. That's why they are putting the mitochondria as part of the double membrane bound organelles. So the prokaryotes, those ones that are not serious, they lack all that. Hope you are together. That is the first distinguishing what? Characteristic. I'm giving whatever statement I'm mentioning time because I'm the same person saying this. And one of my active things is my long-term memory. I can never forget what I say. So trust me, in the future topics, there are things I will not repeat. I will just be referring you to these lessons. That's why I urge you to be taking note of whatever I say. Okay? Now, what is the other thing you realized? You realized that the DNA we have been dealing with is something like this. Just because people didn't get a chance to have further magnification. But you realize that's how the DNA will look like. We shall also see that the other eukaryotes have a different form of DNA. The DNA is just like that. When you look at this thing has two things. The very first thing, it is just one strand. They are not two. It is one complete strand like that. Only one like this. That's why we are calling it single strand of DNA. Then when you look at how it is portrayed, it tries to bring out a circular structure. You get? It tries to be like that, circular. So that's why they are also calling it circular. And even the way I draw it shows that it is only one strand. And it is coming out as what? As circular. So there are two things you really need to know about the DNA of the prokaryotes. One thing, it is single, meaning just one strand of it. And it is also circular for the way it is portrayed. Even when you're illustrating it, you illustrate it the way I've tried to show you. Then you realize that the DNA of eukaryotes, the other ones that are serious, the true ones, the mature ones, their DNA has proteins on them. Has proteins. Hope you're not hearing proteins for the first time. Those proteins are what they call histones. But remember, these ones are for the eukaryotes. Remember, we shall also look at them. So me talking about them before is no problem. So their DNA is just okay because it is covered by histones. But then it turns out that the DNA of the prokaryotes does not have those proteins, but the other one has. So that DNA is called naked. So in assessment, if at all I'm the one to assess you, these are things I really ask you. Why do we say that the DNA of prokaryotes is naked? I want you to tell me that for it, it's not having those, okay, it's not in touch with the histone proteins or just histones, which are proteins, like the eukaryotes, things like that. You have realized they have ribosomes, but their ribosomes are smaller, and we call them the 70S. Most people usually don't know what the S means. The S is the unit that really measures their size. So for these ones, they are 70S. Then the other ones, they are 80S of the eukaryotes. Are we together? So the other ones of the eukaryotes are what? Are larger. And their size is 80S. Then the ribosomes this side, it's like saying that that someone is heavier than the other one. Then we say the other one that is heavier is 50 kilograms. This one that is lighter is okay, the other one that is heavier is 80 kilograms, the other one that is lighter is 70 kilograms. So look at that S as the unit, and then these ones are as the values. Because one of the students in 2023 told me that, but doctor, for us they told us prokaryotes only have 70 ribosomes. So 70 is not about the number, but it's about the size of only one ribosome. That's why they are talking of smaller and larger, things like that. So S is a unit, 70 is the value of the size. Is it okay? Now they are saying that the cell wall, I don't know if people saw the cell wall. You saw that the outer one was the capsule. Then after the capsule, we had the cell wall. Then after the cell wall, we had the plasma membrane. And it is the plasma membrane that was enclosing the what? The cytoplasm, isn't it? So the capsule, this was the cell wall, then this was the plasma membrane, or what you can call the cell membrane. Just, you get. So they are saying that cell wall, the one in the middle, is made up of what we call peptidoglycan, or you can call it moraine. Now, these are biological terms that have no explanation, but you're supposed to get them. Biology is a subject that gives you a word and later gives you the meaning of the word, but not in a meaning faster and hence the word. For example, they tell you that the cell wall is having peptidoglycan, so you're supposed to understand the word peptidoglycan and make sure you do not forget. And they are telling you that the other word that is synonymous to it, murine. You choose what you want. Then biology will proceed and tell you that peptidoglycan has two parts. It's having the peptido part, which will represent protein, and all this shall be looked at under chemicals of life. No need to worry. And then it's having the glycan part. Yes, it's having the glycan part. Glycan part, people have heard of glycogen, and you all know glycogen is a carbohydrate. So every time you see that glyc, things like that, they really mean carbohydrate. Carbohydrate. Then I know whoever is here knows that peptide is related to protein because proteins are polypeptides. I don't know if people are hearing this for the first time, but you will learn, don't worry. Yeah? Okay, at least people know that proteins, when they are broken down, they give you amino acids. Ivan, leave our screen. So what joins amino acids together to come up with the protein, they are called peptide bonds or peptide links. So peptide and protein, they are just together. Just like you can see glycogen, that is a carbohydrate, usually means carbohydrate. Yeah? So what I can say right now is that the peptidoglycan, that is a component of the cell wall, is a protein carbohydrate complex. Otherwise, have it in mind that the carbohydrate in this case is a polysaccharide. Carbohydrates are not part of your syllabus, and it was very bad because you need them so much. But I don't know if the people here are aware that carbohydrates are composed of 3 types or classes: the monosaccharides with 1 sugar unit, disaccharides with 2 sugar units, and the polysaccharides with so many or 3 or more sugar units. That's what I told you, that you really need to have a book and pen, because I'm saying a lot than we actually have, or I'm going into other topics. Like now, have you noted that carbohydrates have 3 classes? Do you have the word monosaccharide, disaccharide, polysaccharide? Have you put in brackets single sugar unit, double sugar unit, and multiple sugar unit, 3 or more? Do you have those things? So the glycan here is strictly a polysaccharide with a name mucopolysaccharide. And I'm going to explain to you why we bring in the word muco later on. But of course, in biology, when you see muco, you think of mucus. So it is simply those polysaccharides in mucus. And you know we have mucus everywhere, in the intestines, everywhere, nasal sinuses and everything. And all that information is in textbooks of your level. I told you it is very hard for you to pass this subject if at all you don't read textbooks. But make sure these basics we discuss become part of you slowly and slowly and slowly. By the time we complex the content, you'll be understanding more. Are we together? So you can just see just one point. Peptidoglycan murine, a lot has been said. Hey. Okay. What is the other point? Distinguishing characteristic. They're saying extremely small cells, approximately 0.5 to 5 micrometers in length. Very important value. But you guys don't need this value. It was needed in the old curriculum where they used to set objectives. But as of now, you just need to know that they're extremely small. That is enough. If you can remember the estimate, the approximate, well and good. You can recall it. Okay? I think we can proceed. Let's handle the second note. Okay, what are they saying? They are saying that the prokaryotes, propro means before life. They were there. They were there. If at all they were not there, okay, they were the first. That's why they are calling them the earliest, the simplest, and most primitive. I don't know what they currently handle in history of the new curriculum or level, but those days they used to study the people they called the Sans and Khoisans in South Africa, and they called them the very first people who inhabited the country. And because they were the earliest who inhabited the country, they used to call them so primitive. You know, primitivity means something that is backward. You understand? So by backward, we mean that maybe that something has been there for some good time. Maybe it was the earliest, so they call it most primitive. So by most primitive, we mean something has really been there, or it is even too backward. The mere fact it was there too early. And since it was there too early and things keep on advancing, that means it is the simplest. Because as time goes by, we move from simple to complex. So hope you get the early part, the simple part, and the most primitive part. Okay? Now, they are bringing in a terminology here, and they are saying the chromosome. Chromosome. Now, chromosome and so on is something that really gave people hard time. I think I was introducing cell division, and that is one of the topics you're supposed to cover in Senior 6. And I'm not going to say much about a chromosome, but all I can say, a chromosome is related to DNA as of now. But the lesson, I've told you the lesson that really explains the exact relationship between a chromosome and DNA. It's the lesson of cell division. You get? Yeah? So they are saying that those DNA strands you saw, I think you saw them, they are coiled up in the center. Remember those red things you saw? They coil up in the center. And what they form is what they call a nucleoid, and I told you about that. Hey, you people, this biology. Membrane, peptidoglycan, now nucleoid, you're about to see mesosome, you saw 70S. A lot of things are still coming. You saw plasmid. Some of you have just had ribosome for the first time, and you're still hearing. The ribosomes are those small things you saw inside the cell. They are responsible for protein synthesis. That small thing you saw, the ribosome, it's the one that is responsible for protein synthesis. Hey, you should know that. Let me try to show people, before we even proceed, something about the structure. Now that's the bacteria you've been seeing. Are you seeing these small things? I told you the single one is the pillars. The plural is pilli or pilli. Are you seeing the capsule is the one that is outermost, if you can realize that one, outermost. Then the next thing is the cell wall, that one. Then lastly is the plasma membrane. I don't know if people can see it is very thin. And that is one of the characteristics of the plasma membrane. It is known for being very thin. Are we together? Then you all know immediately after it, that's where we find the cytoplasm, gel-like fluid and so on. Are you seeing the plasmid? I told you should remember that the plasmid is ring-like. See? Are you seeing its DNA? So DNA, I told you it's connected to the word chromosome. Chromosome simply means DNA strand. You can see it is coiled up, and that thing is one strand, and you can see the cellular thing I told you about. Just because now the coils are too many, but it is a single strand, and you can see the cellular thing there also. So the ribosomes are those small things you see, very small. And those ones are the ones where protein synthesis occurs. But I can surprise you that they are too big in that they have two subunits. Just because for now we just need them as them. Ribosomes, nothing else. These longer ones, I told you one is flagellum, singular flagella. And I told you they are responsible for locomotion. You see? Then these are just some other inclusions, and we are going to see them. There are so many inclusions. As you hear the word inclusion, they are included within the cytoplasm. So the other one that I had not talked about is the mesosome. So the mesosome is just usually attached to the plasma membrane. It can also be like this. It can also be like that. It can have so many structures, but have it in mind that it is weird. Yes, it's always having different structures, but attached to the membrane. Now, you need to know two things here. That in most of the cases, we have those structures that are always present on a bacterium cell, and then those that may or may not be present on that cell. You encourage people to log on. I don't know why people are disappearing, but the number that is here is very okay. I like that. Your doctors are supposed to persevere and endure. Even if the parent tells you to sleep, don't sleep. It is your future, not theirs. and I'm saying this boldly. Yeah, tell them, let me struggle for the 2 years. I will rectify my mental health after passing. And the happiness you get after passing is enough to reveal and revive your mental health. And why would you lose it if you're covering work ahead of teachers? You're just very safe. Is it okay? Now, we are going to see some other structures. There are so many. There are also the photosynthetic membranes. They are also always parts. They also always try to project from inside or from within the plasma membrane. So you realize that this bacterium cell does not have the mitochondrion where respiration can occur. So respiration, strictly aerobic, occurs at those membranes. They call them mesosomes. So you're just learning new words. I don't know if that is true. But now mesosome is another new word. And you should be aware that there are membranes where respiration is going to occur because our bacterial cell does not have the mitochondrion where aerobic respiration would occur. About aerobic and anaerobic, we shall see that under the topic respiration. It is topic 4. Hope we are together. Okay. So we can go back and read. Where are we? So I think you realize the nucleoid, that coiling up, the group of the coils in the center, it's the nucleoid. They are saying that in addition, there are many smaller rings of DNA called plasmids. So those plasmids you've been seeing, the diagram had only one, but there are already so many. And they are smaller rings of DNA. Why? We already said DNA is a circular ring. So that means for it is very large. But these ones are smaller and there are very many. And they are the ones we call plasmids. How do they work? They work as accessory chromosomes. I told you chromosomes, I told you the video to watch. So by accessory chromosomes, we are going to see this as we talk about the function. But when they tell you accessory is just something that helps you reach something, but it is not so important. You may have power. You may have, I don't know that people know cables. You may have a cable, but then you lack an adapter to charge your phone via USB. That's why they call those things accessories. If you have an adapter but you don't have a USB to charge a phone, now this thing, the USB is an accessory. It just helps you to access the electricity. So the plasmids don't really do what we may expect by the pass access DNA, so that they can control certain things, as we are going to see. But have it in mind, plasmids are a very common point of assessment. They like them so much. But all you need to know, they are really smaller rings of DNA. And the accessory part, I've told you, they may not do exactly what you may think, but they help us access what DNA can do. After all, chromosome, the word is related to DNA. Is that hard? Okay. Now, let us read this statement that tries to summarize a lot of content. They're saying that the cytoplasm contains few organelles. That means the eukaryotic cytoplasm has so many. This is for the prokaryotes. And they are saying that, and in none is surrounded by a two-membrane envelope. I told you about that. You saw the two membranes on both the mitochondria and the nucleus. They are talking about the 70S ribosomes again. They are talking of the storage granules and the enzymes. Is it okay? Someone may be like, which storage granules exactly? We are going to see all of them. Enzymes, you had that from the video. You get. Then, the other thing they are talking about, there's the thing they lack. They also lack membrane-bound organelles. They're just saying membrane-bound. Leave alone the other thing of double membrane. This one is just membrane-bound. Someone may be like, what do you mean ribosomes don't have a membrane? Yes. The ribosomes, you see, they're just there as ribosomes. It's not like they have a membrane on them. They don't. So even the organelles that just have a single membrane, these prokaryotes don't have. They don't. Those things you said just inclusions, and the other, they're just the membranes, as we're going to see, the mesosomes and what. But you see, they are talking of mitochondria, chloroplast, they don't have. Endoplasmic reticulum, they don't have. So you should learn. These are now all organelles. Someone may be like, what is that endoplasmic reticulum? We shall see it in detail under eukaryotic cell. We are first talking about these ones, so they lack. First have that in mind. So just learn now one thing, that prokaryotes, they not only lack organelles with a double membrane, but even those with a single membrane, they lack. Then when we reach the ultrastructure of the plant and animal cells, we shall see some of these in details, and we shall watch a video. You can even just go to YouTube, put in 3D animation of the endoplasmic reticulum. You have a look at it. Trust me, you can't remain the same. The others, we have what we call the lysosomes. We shall see why they are called like that. We have vesicles. As we hear the word vesicle, vesicle is something with a fluid inside in most of the cases. It's just an English word, not so related to science. Then we shall also see the Golgi bodies, or what they call the Golgi apparatus, what they call the Golgi complex. And then they also have some other name that is too scientific, and I will give it to you in the future. So as I told you that they don't have the mitochondria where respiration, strictly aerobic, can take place. So who takes over the role? The mesosomes. And I told you they're just inward projections of membranes from the plasma membrane, still within the cytoplasm, as you can see. Hope we are together. I told you you're supposed to read these notes immediately after this class or tomorrow morning, so that you really recall very fast whatever I said, so that you attach the notes. Attach the notes like that. Okay? Now again, about the structure. Yeah? So, this side, we have the structures that are always present. Always. You see that? Always present. Then here we are having additional structures. Sometimes present. They're just additional. So that means they may or they may not be present. But these ones, they are always, so these ones must be present. So this is what can really bring out what I may want. Okay? Now, let's first begin with those that should be there. Yeah? Let's first begin with those that must be part of everything. So after this, I think the next category is the functions. Now, this is where biology is. This is where biology is. Because now we are done with structure. So the next thing is supposed to be function. And function is in form of a table. So when we get done with this table, we shall be done. Yeah? These other things are actually your research. Yeah? They're just your research. It is the table that is concluding this class. So make sure you look at this structure and understand. But you already know whatever is here. We are just making the point of what is always present, what may or may not be present. Okay? So I'm not interested in the functions. You see? So the outside thing that is, how can I say? The most outside or the outermost layer is the capsule. It's here. As you can see, it is here. This one. And they are saying additional protection, but we are going to read the notes. This diagram is just there to give us what we need. Then you can see after the capsule, we have this, the cell wall. You see it? Then after that, we have the cell surface membrane. I think this book is very bad. It's very bad. It didn't label the stuff very well. I don't like that. The cell surface membrane is this. Are you seeing that color in? Because it is a membrane. So those two are always present. It is the capsule that may or may not. Are we together? Let me change ink. Then, do you see the plasmid? It is inside the cytoplasm and it is a ring. And you should be knowing that it is a ring of what? DNA. Are we together? Then you see the ribosomes. The diagram is not very good and I had not rectified that. I'm really sorry. But the ribosomes are these ones. You see the cellular DNA. You know it is single-stranded and cellular. Told you chromosome is related to DNA. Then you see the pilli. You know the pilli, they are also not always present. You know they are always just there. Even if you just go to Pinterest and put in image showing the structures of a bacterium cell that are always present and the additional structures, they will give you a very good illustration. And trust me, you'll get it. Then this one is the flagellum, the flagella, and it is responsible for what? Locomotion and movement. Hope people know that movement. If I told you you're doing break dance, you're in one position but you're moving your hand along, that is movement. Movement of part of an organism. Then locomotion, it is the whole body moving, like when you're running. If you just nod your head, you're just moving part of your body, that is movement. Hope you know that difference from the characteristics of living thing. So this membrane you're seeing here, this one, I think let me use blue. That could be the mesosome. I read it's just an infolding of the cell membrane, infolding. You see? So they are telling you that it could be a mesosome for respiration, it could be a membrane for nitrogen fixation, it could be a membrane for photosynthesis. So all those membranes are there. But the most common mesosome and those of photosynthesis. So you should be aware about the structure. So a lot has been said about the structures. We are now going to look at the part, specific part, and the function. I believe some of the people are here are part of the biology scholars group in the SSA community. So please, you tell people in the biology scholars group to log on. You send them the link, let them at least come back and appreciate something about the what? The function and structure. Structure is done. Let them come and appreciate more about the structure and then the function. The moment we finish this table, I'm going to just give you the story about the division of kingdom Monera. Then I'll give you research for you to go and tell me about the eubacteria and archibacteria. Yeah. Then we shall end. So this is just a summary. Then these differences, they are used. Then these other things you can even get from the internet. And then the next thing shall be eukaryotes. And eukaryotes, according to the plan for today, it's our take 2. That means this will be next time, the eukaryotes. That is okay. Now, to go back. So tell your friends to come and listen to the content. Here is our table. Okay. So we are going to look at the part of here we are looking at part of the prokaryotic cell and the function. You now know all parts now. We are just relating. So they are saying that the cell wall contains murine or peptidoglycan. And you all know that the cell wall is usually important for protection. You get, because they're telling you that the function is, it is a physical barrier that protects the cell against mechanical damage. That means this cell wall needs a strengthening material for it to be in position to be a physical barrier to protect the cell against mechanical damage. And it is the murine, or what they call peptidoglycan, that is the main strengthening material to really make it a perfect physical barrier. So being a physical barrier, it even helps in excluding certain substances. Maybe a substance that can really kill the cell wants to enter. The peptidoglycan works as a physical barrier and that thing does not what? Does not enter. For example, if water enters a cell, it swells and bursts. So this, we are saying that this peptidoglycan also helps the cell via the cell wall not to burst. So we are saying that it prevents bursting in dilute conditions. We shall see that in osmosis, that when you put something in a dilute condition or in a dilute solution, it means that it is surrounded by a lot of water. So that a lot of water may be taken in, and when it's taken in, the cell may increase in size, swell until it what? It bursts. So because the murine is giving this cell wall main strength, bursting will be prevented because swelling will not happen. And the mere fact that water is not entering to make this cell swell and burst, that means its shape is also going to maintain. The shape is going to maintain. It's not going to change. And by the mere fact all that is being done, protection from external damage, certain substances are excluded from entering, shape is being maintained, you are more or less supporting the cell. So those are all functions. So I told you peptido means protein, and we connected it to polypeptide. We shall see those terms in the future. But as of now, understand protein and know that proteins are made up of amino acids. So when two amino acids combine, it becomes a dipeptide. And I told you what connects two amino acids is what they call a peptide bond or peptide link. When three amino acids come together, it's what they call a tripeptide. When four or more amino acids get together, it's what they call a polypeptide. And you all know many amino acids together, they make up a protein. That's what I meant. And I remember telling you that glycan is polysaccharide, but strictly what they call the mucopolysaccharide. Hope we are together. Very important work. Okay. We go next. The capsule. The capsule was on the side of those that may or may not be present. That's why they are telling it is in some bacteria and it is slimy. By the mere fact it is slimy, that means it's having some liquid. If it's having some liquid, that means it will prevent the cell from what? From drying out. Because it is slim, it has that liquid. Trust me, the cell won't what? Won't dry out. And the mere fact it is slimy, the bacteria will be in position to stick together for further protection. Yes, they can decide to just clamp together. You can protect yourself as an individual, you can get into a group and protect yourself with combined energy. So the capsule being slimy, it can stick together. You all know most of the things that help in sticking are slimy. And there is also extra protection. It is a capsule. It is covering the cell wall, covering the plasma membrane, covering the cytoplasm and all the cell organelles. So the protection is just extra because we know the main guy that does the protection thing is the cell wall. So never forget the word extra. So they are saying that from other cells, they are talking of the leukocytes. We shall see that under transport in humans, strictly defense and immunity. No need to really bother about them so much. It's good to talk about leukocytes when we are talking about lymphocytes, at the same time erythrocytes, then the leukocytes. There are so many, but I know people know them. At least at your level, you should be knowing that the erythrocytes are the red blood cells and the leukocytes are the white blood cells. You know what red blood cells do? They are really responsible for transport of oxygen mainly. Then the white blood cells are responsible for defense against diseases or those causative agents of diseases. So they really always fight cells. And don't you know that bacteria cause diseases? So that means they are causative agents. So leukocytes, the white blood cells, are supposed to fight against those bacteria. So the capsules can provide extra protection from cells, which are the leukocytes. I don't know if people connect the biology. A lot has been said and not written. Hope you've noted or taken note. Okay. Flagellum. Of course, it was also on the side of being present in some but not all. So they are saying that the flagellum is lacking a 9+2 pattern. So this 9+2 pattern, you see most of these things where we say lack, we shall talk about them in eukaryotes because the eukaryotes have. We cannot talk about them if at all they are lacking. It makes sense to talk about where they are present, but don't you worry. And we are saying they also have, or they are just hollow cylinders, just like the way eukaryotes are. And remember, we shall watch the videos, we shall read the content, and everything shall be coming out. And what is its role? Movement and locomotion by wave-like action. You saw the video, you saw the wave-like action. Okay? Now we have the pilli, and I told you the fimbri. I told you that name too, so you choose what you want. The singular is pillars. It is also part of one of our diagrams, pillars. And it is also found in some cells. We said it is important for attachment. I said that already, and sexual reproduction. All this was already talked about. The cell membrane, we talked about it being selectively permeable, in that it regulates what enters and exits. Yes. And remember, we said from the cell membrane, we have enfoldings that may form things like the mesosome, which mesosome is a site of respiration. That means in one way or the other, this cell membrane of strictly the prokaryotes can be a site of respiratory enzymes. Then the mesosome is a site for aerobic respiration. How? They are continuing and telling me that it provides a large surface area for the attachment of respiratory enzymes, if you want to know how. But you all know if this is the cell membrane, the mesosome, I told you it comes off like that. So those things really provide the large surface area they are talking about. And you should be knowing that because of the way it projects in one way or the other, it may come in close contact with the DNA, something like that. And being in close contact with DNA, that means it is associated with what? With DNA. It can really get close to it. And this only happens during cell division, and we end up forming new cell walls. Now, cell wall formation is talked about somewhere, and that is in plant cells. But the topic where it's talked about very well was removed from your curriculum. But for the mesosome, just have it in mind, site for aerobic respiration period. The large surface area I've showed you how it comes about. Those projections try to get close to the DNA. So anytime the cell divides, they may associate. And in the due course, new cell walls are formed. So you can go to YouTube and put in how is a cell wall of a bacterium cell synthesized. You watch the video and you see the content coming through. This biology is a subject where you must build your personal content, you as the person. Yeah? Okay. Ribosomes 70S type, you know that, and it is a site for protein synthesis. Now, I told you about these membranes. They are like the mesosomes. They project their enfoldings of the plasma membrane. You see? So you can call them photosynthetic membranes, photosynthetic lamella, or simply the thylakoids. And we shall see this under structure of a chloroplast. You see what lamella means, what thylakoids mean, things like that. And they're also telling you they are found in some and not all. And some people just simply call them chromatophores. But for now, just have photosynthetic membrane. I'm advising you as the tutor. Chromatophores. Why chromato? Because they are colored. They are simply rated to color, nothing else. We shall see that when we reach about the archaea and eubacteria. We shall see the colors as I'm trying to say here. But as of now, just have them as photosynthetic what? Membranes. Chromatophore simply means, chromato is rated to color. I don't know if people know chromatography, paper chromatography or just chromatography. It's related to separation of colored what? Substances. And you all know photosynthesis one of the things that are responsible for it is a green pigment called chlorophyll. So chlorophyll, green pigment, you can see the color coming in. Hope we are together. So they are even quoting it and saying that these ones do what? Contain pigments. And since we are dealing with bacteria, the pigment of bacteria, this is now another new term you're learning today. It's called bacteriochlorophyll. Bacteriochlorophyll, very important. It also works like chlorophyll by trapping what? Light energy. What is that light energy needed for? Photosynthesis. Photo for light synthesis. Is it okay? Now, this note is concerning other species of bacteria, and I think it's going to be talked about still under the Archaea and Eubacteria, because at least I remember it, because it's me who organizes these notes. Because I remember it wasn't part, then I added it. But when we talk about the Eu and Archaea bacteria, it will make sense. Okay, we proceed. Cellular DNA, you know what this one does? It's the one that really handles or keeps the genetic information for the replication of bacterial cells. You know, when replication happens, it's like parents are giving you daughter cells. And we talked of DNA being the form of transfer, or the mid-zone mode of transfer of traits from parents to daughter cells. Hope you all recall that. So we go to the plasmids. Plasmids, we said they are small rings of DNA. You all know that DNA always replicates. We cover that in nucleic acids. So when they say self-replicating, that means they can replicate on their own without really getting any force, any motivation. They can just replicate themselves. And trust me, there is a lot of content under plasmids. A lot of content. That's why they really end this all. And I want you to listen very extra carefully. So the mere fact that they are containing DNA, or since they are DNA, that means they have genes. I don't know whether people are aware that a gene is a section of DNA. These things really don't make sense. You usually just cram them that plasmid is a ring of DNA. Then it has genes. But when that topic of nucleic acids is covered, you can at least cover and go and look at the structure. But what I can say is that, or what I can tell you now is that DNA is made up of genes. And I'm going to just tell you a story, just get it locally. Why they always call it a DNA test? Because they believe the DNA has genes and they want to check whether the genes of this child are the same as the genes of the father. Do you understand? I think it's now okay. DNA has genes, so they call it a DNA test because they want to check, is this DNA having the same genes? Is this DNA of the child having same genes as those of the father? Period. So what do these ones really do? What do these genes really do? They just give extra survival advantage in bacteria. As simple as that. And they are saying where they are found, so even the plasmids are not everywhere. I don't know, for them they just play an accessory role. So they are not so important as you may think. But where they are present, they do the needful. And we have told you, you all know they are small rings of DNA. And because they can replicate, that means that is so good of them. And then they possess some genes, because we expect DNA to have genes. But we have said the genes are very few. But that's not a concern. So now the thing is, how are these genes really going to help us? Now there are so many applications that are included in this explanation. But you also need to get it, because you're students of biology. So what are they saying? They're saying that these genes play a very important role, and that role is really about extra survival. So most of the things we are going to be listing are in line with survival. So plasmid, survival. Whichever thing we write under it is about survival. A case in point, They confer resistance to antibiotics, and I will first stop there. I'm not going to say all this because people will be like, things are now getting serious. But have it in mind, all this information was gotten from your reference books of the new curriculum or of your level. So nothing is complex here. Someone may be like, Anna, what is this thing of the staphylococci? There are things that are taught in even primary, if you recall. Streptococci, staphylococci, there are things that are talked about. They're just forms of bacteria, but they shouldn't really scare you as you're reading. Don't. If at all this is hard, remove it and say some bacteria. But staphylococci is just a class of bacteria, of course related to shape. You know, there are those that are rod-like, there are those that are in clusters, there are those that are in chains. From primary. For me, I'm recalling all that from primary, not even A level. That bacteria, according to shapes, there is rod-like shaped bacteria, there is bacteria that is in clusters, there is bacteria that is in chains. And I know the one in chains is the streptococci. I know the one in clusters like grapes is the staphylococcus. And then the one that is rod-like is the bacilli. That is primary, and that was covered in science. I don't know which topic it was, but we used to look at those things. Back then, I was in primary, I think even before 2010, long time ago. Okay? Now, so they have been talking about some bacteria that has a certain plasmid, so on and so on. It is related to penicillin. Okay, let me talk about it, because you need to learn these things. At least you leave the class when you've learned something very hard. So they are saying there is some bacteria that has a plasmid. That plasmid has a gene, because a plasmid is DNA and DNA has genes. That gene helps us to make the enzyme penicillase. And you all know when you hear something like maltase, it breaks down maltose. Sucrase, it breaks down sucrose. I think you people know that. Lipase breaks down lipids. Peptidase breaks down peptides. So the enzyme penicillase, we expect it to break down what? Penicillin. Now, penicillin is an antibiotic. You learned that from me. By antibiotic, biotic means life. Anti means against. So an antibiotic is a chemical that is against life. So penicillin is a drug that can be used against the life of bacteria. So certain bacteria have plasmids which have genes, which genes help us to make enzyme penicillase that can break down penicillin. So the penicillin cannot affect the bacteria. So the bacteria is hence resistant to the penicillin, the drug. Hope we are together. And how can those genes be transferred? By conjugation. Conjugation is a form of sexual reproduction in the bacteria. Are you seeing there are a lot of content? That's what makes biology hard. That there are a lot of things prone to being forgotten, but you must understand. Repeat and repeat them over and over until the point is driven home. So a lot of content is there. I was even going to skip it, but I said, no, let me say it. So some bacteria have plasmids. Those plasmids, since they are DNA, they have a gene. The gene will help us to make enzyme penicillase. Penicillase, as we are, penicillase is supposed to break down penicillin. Penicillin is an antibiotic. That means it is against life, but against life of what? Of bacteria. So if penicillase is made and penicillin is broken down, that means the penicillin cannot affect the bacteria. So the bacteria is resistant to it because of the penicillase made that breaks it down from the toxic form to the non-toxic form. How can those genes be passed on to other bacteria? By conjugation. What is conjugation? Is a form of sexual reproduction in bacteria. Okay? I think let me hear from this person, Cyphers. Yes, the moment you see people reducing in a biology lesson, you just know now content is becoming too much and people are running for their life. Okay? I don't know, the person who is up their hand is not talking. Maybe I can proceed, they will talk. We have confer resistance to disinfectants. Disinfectants are like antibiotics. They're just there to kill bacteria. So the resistance also comes in. Allow bacteria to cause disease because they are being resistant to things that kill them. So plasmid just give them a chance to really still cause diseases. You get? And we have we have a form of bacteria called the lactic acid bacteria. It is responsible for fermentation. You understand this more when you reach the topic called respiration. I told you this topic is hard in that it can really make you feel like the head is becoming too heavy because of taking in a lot of content. Because it brings a lot of examples from so many topics in biology. So, the mere fact the lactic acid bacteria is not dying, it can keep doing the fermentation. So, then the other thing, since we are breaking down things, in the industry, they usually make what they call hydrocarbons. I taught you hydrocarbons in organic chemistry. How they are used as clearing agents in oil spills. That is just English, not science. So, these hydrocarbons can be broken down because they are very complex. So, they can be broken down and bacteria can use them as food. Then we also have petroleum. Petroleum can also be broken down into proteins, and these proteins can be used as food. So the petroleum, the hydrocarbons are complex chemicals. So those plasmids have genes that can really give bacteria the ability to break down those complex chemicals, hydrocarbons and petroleum, into food or into things that can be had as food. Hope we are together. So you just need to make sure these things make sense. Then it is you to sit down and memorize after reading for the second time. Okay? Now, I remember telling you about the inclusions. Inclusion, the other things that may be included in the cytoplasm. And here we go. They're here. We have the lipid droplets, we have the glycogen granules. As we hear the word lipid droplets, they are simply supposed to store lipids, nothing else. Glycogen granules, I told you glycogen is a carbohydrate, so they're supposed to store what? Carbohydrates. And you know carbohydrates are broken down in respiration to provide energy. Glucose is a carbohydrate. Don't we break it down in respiration to release energy? Yes. Just because here they are adding on some other things. Yeah. That lipid droplets, they store lipids as more concentrated and long term. So they store lipids for some good time, and it's where a concentration of lipids happens. And when they store later on, they can convert it to carbohydrate if at all there is need for them to be respired to release energy. So they store lipids. But later on, if need for respiration arises and the carbohydrates are not enough, those lipids can be converted to carbohydrates and then respiration proceeds. That's what they are meaning. That's what they are meaning. Okay. Now, whatever you're seeing in these notes is in line with the curriculum. It's what the curriculum really urged you to be knowing. So especially these other points, they are the ones that make you have extraordinary content that is not known by anyone, but good content. You can see they are saying some prokaryotic cells like I don't know whether people can read that. I don't know whether people know this SPP. I'm going to give you this as research. Yeah. You people find out what I mean by SPP. But if at all you cannot recall the anabana, I'll say anabana. Yes, I'll say anabana. If at all you cannot recall the anabana kind of species of bacteria or prokaryotic cells, for you just have it in mind that some prokaryotic cells have a membrane for fixation of atmospheric nitrogen into organic compounds that are strictly amino acids. So in chemicals of life, you see what amino acids are. Yeah. And I think I told you that already in our diagram. We say those infoldings are either membranes for nitrogen fixation, membranes for photosynthesis, or mesosomes. Is it okay? Now, this is where the thing is. Yeah? They are saying that prokaryotes belong to kingdom Monera. Kingdom Monera. And kingdom Monera consists of all bacteria and cyanobacteria. Are we together? You know those things of, you remember the mnemonic? We used to have those days of senior one. We used to say that kings play cards on flat green stools. Kingdom, phylum, class, order, family, genus, species. So the highest is kingdom, and then the lowest is species. So they are saying that the prokaryotes we've been talking about, they belong to kingdom what? Monera. And what do we have there? We not only have bacteria, but we also have what we call cyanobacteria. So things like cyanobacteria, they are the things as you're taking home as research, because now more serious content is there. And the content under cyanobacteria is not ordinary. It is advanced content. You're going to take it as research, but I count it as something that is going to be part of our lesson too. Yeah? But I'm going to first give you a hint about kingdom Monera. Then you go and make research about your bacteria, Archibacteria. You have textbooks, all of them. They're even present on the free drive link. I want you to first make some research, or you take it as research for school. In the first week, make it take to the teacher to confirm. But I'll also be here. I'm also posting the consultation line of biology after the class. I just need people to remind me. Is it okay? Now, you all know that from kingdom, we go to phylum. From kingdom, we go to phylum. These are the levels of classification. I know that people know taxonomy. I don't know if they have heard of taxonomic classes. Kingdom, phylum. That's why they are making a statement here. The plural of phylum is phyla. That's why they are saying that kingdom Monera is subdivided into two main phyla. Phyla. They are talking of phyla. So, what are the two phyla? The statement is there clear. Bacteria and cyanobacteria. So the phylum is cyanobacteria, that is one. The other one is just phylum bacteria. Now, you're going to get more content from here, but don't burst. Please don't burst. To begin with, let me ask you people. I think you've heard of cyan, the color. You see, you people, we have not had something or anything written in the workbook. We are just simply using our notes and following up. So, you've heard of the color cyan. Someone tell me. What are the colors in cyan? That's what I want to know. Cyan, the color. Because we are saying cyanobacteria. So that means something should be related to color. So who knows the colors? Cyanobacteria. Okay, cyan, blue and green. Someone is saying there are so many unfamiliar terms. You're lying. At least all unfamiliar terms we are coming across, we are trying to say something about them. Is there any word? I think people in the chat can really bear me witness. Is there any unfamiliar term I've not talked about? At least I try to tell you that. What I see that may take our time and may give you time, I tell you to first leave it. But I think everything we're endeavoring to say something about it and left clear. Isn't it? The answer is blue and green, but now I need support. Someone is claiming in the chat that there are so many unfamiliar terms. But you're supposed to learn them. You're supposed to learn them. Because now your syllabus brought in something new, the kingdom Monera. And since it is new, I'm going to give you a hint. I leave you with assignment. Still under Monera. Yes. Okay, so please don't burst. So people are saying that cyan is blue and green, and that is true. Blue and green. Okay, we go. That is enough. That is enough to read this paragraph and understand. Okay, we go. These are mainly spherical or rod-shaped. Are you seeing? Cells of unicellular structure. So by unicellular, it means they are single-celled. And they are saying they are mainly spherical, but they can also be what? Rod-shaped. You know, spherical and rod-shaped are kind of similar, but not really that. You know, a sphere is like this, like a ball, then the rods are like this. You get? So there is a way they try to look similar, but they are not really similar. Just slightly. You get? And they are saying they are arranged in simple or branched multicellular filaments. Now, these are characteristics. These characteristics were so important in the old curriculum where they used to set objectives. They say which one of the following is a characteristic of cyanobacteria, or the following are characteristics of cyanobacteria except. So these things shouldn't overstress you. But I'll always quote out something you should really remember. So someone may look at these things of spherical rod shapes, unicellular, again, simple branch multicellular filaments as something hard. But remember, it was extracted from biological science, which is your textbook, and the curriculum wanted you to know the subdivisions of kingdom Monera. It's here. Read. Explain the division of the original kingdom Monera. So the original division was this one of phylum Cyanobacteria and phylum Bacteria. But then there are the new kingdoms, the Eubacteria and the Archaebacteria, which you're taking home as your assignment. Hey, hope we are together. So we are talking of the old divisions. Then you people are going to teach us the new ones. Okay? So what should we really note about the cyano? I told you that every time you see the word photosynthetics, think about color. Because the photosynthetic pigments are colored. Are we together? And cyano, I've told you from cyan, the blue and green, it's related to color. So don't be surprised when they tell you that they are photosynthetic prokaryotes. And they have photosynthetic pigments. And these photosynthetic pigments, you all know they are intact with the plasma membrane. They are enfoldings. This is the word that we have as enfoldings on our diagram. And they are saying around the periphery of the cell. I know that people know the center and the periphery. I also taught this under coordination under the nervous system, because what we have what we call the central nervous system and the peripheral nervous system. So periphery is like away from the cell. Okay, people, this is it. If at all this is a cell and this is the nucleus in the center, so this is the center of the cell. Now here around where the cell is ending, where I'm shading, that's what they call the periphery of the cell. And you all know it is from that area where we had enfoldings, the interkings that came through as the photosynthetic pigments, if you recall. Now, they are trying to bring in, they are telling you that the photosynthetic pigments, they're now talking about them. The photosynthetic pigments are three. The first one is bacterial chlorophyll. Please, bacterial, you should understand that it is O. I don't know if people know that in biology that thing works so much. You can have dorsal, then you can also have the word ventral. But if you at all you want to combine them, this dorsal, this one, becomes dorsoventral like this. Just because your curriculum doesn't have the cockroach, but the cockroach had a lot of features on its dorsoventral view, and they were so important in its adaptations, but that is bygone. So as you're writing bacterial chlorophyll, don't forget to remove the AL and put the O, because you're bringing things together. There's also what they call the bacteriophage. You also see it in biology. What makes biology hard at A level? But people pass, but not all of them pass. Those who pass are people who will repeat it over and over, over and over. It has a lot of terminologies which you may not need. It has a lot of words and facts which you may not need, but you're supposed to hear of them at least for once. And you're supposed to just appreciate. And don't mind they are not everywhere, not in all topics. But this topic is just wordy and carries a lot of lugambo from other topics to it. Yet it is in the introduction. But everything is really worth it. Okay, so the bacterial chlorophyll is green in color. You people told me cyan means blue and green. Then we have the phycocyanin. Is also photosynthetic pigment. It's for it blue in color. Are we together? Bacterial chlorophyll is green. Phycocyanin is blue. Then we have the one that is red. The one they call the phycoerythrin. You people, I told you erythrocytes are red blood cells. Every time you see erythri, it really relates to red in biology. So you should now close your eyes and look up. Before you sleep, you say bacterial chlorophyll green, phycocyanin blue, phycoerythrin red. You get the point? Hey, phycoerythrin red, phycocyanin blue, bacterial chlorophyll green. Is that really hard? So they are saying that some other species of cyanobacteria, there are others that live mutualistically with fungi to form lichens. Now this is another new term, lichen, that we taught under ecology, the very last topic in your biology. I don't know if people are aware that ecology is the 9th topic. Phycocyanin, phycoerythrin, bacterial chlorophyll green, phycocyanin blue, phycoerythrin red. Very nice words. Now what will show that you are an A-level student of biology? Those are the words. You should say them and scare people. But you should make sure that as you're scaring people, you read hard to pass. Because if you don't pass, they will talk about you badly after. So you should be reading and making sure that you're going to pass. So that you don't give a chance to people who are going to talk about everything. Okay? Now, we are saying that other species, mutualism is a form of symbiosis. Those two words usually disturb people of biology. Mutualism and symbiosis. But mutualism is a form of symbiosis. Symbiosis is simply an interaction of organisms living together. Then for mutualism, both should be benefiting from each other. So when bacteria and fungi live together mutualistically, we mean that they are benefiting from each other. Simple as that. And the interaction is what they call a lichen in science. Just like the interaction of a boy and girl or any man and a woman, it's called a relationship. So even in science, a connection of two organisms living mutualistically together, especially if fungi is involved, they call it a lichen. That's why even fungi and algae is a lichen. Are we together? Okay. Many cyanobacteria species contain gas vacuoles. I think I got this from, was it Principles and Applications or Michael Kent? They say that many species they have gas vacuoles. So when these gas vacuoles, as we are the word, they are vacuoles that are supposed to be filled with gas. So when they are filled with gas, they can float on what? On the surface of water in which they live. Just one statement, a fact. Do you really lose anything? And they are telling you that these cyanobacteria, one time they were called the blue-green what? Algae. And they were under the plant kingdom. That's why the word algae. And blue-green brings out the cyano thing. So that shouldn't be anything like a confusion. Those who know English, you know what they mean by that erroneously. I don't want to say much about it. So some or many have the gas vacuoles. They can be filled with gas and they can float. Don't you know that? Physics. PCB students. Okay. Now, kingdom Monera originally was divided into two. Phyla, the cyanobacteria that we have talked about, and the just bacteria. So this one, unicellular. This one, we have talked about it so much. So I've told you that we can have, okay, bacteria can take on different shapes. I told you about the clumps, the ones I called the clusters. You go to Pinterest and put in different shapes of bacteria. You see the one of clusters. It's the staphylococcus, or cocci for plural. Then it can be in chains, the streptococci. You can also go and see. We have those that are rod-like. So many, even those in form of simple filaments. Okay, that's about shapes, different shapes, but it's not a big deal. Okay, and you had those things in primary, so I don't think I'm really saying things that are too much, but you should be taking note. But thank God we are done with the class. Hey, we are done with the class. We are done with the class. Now, I want people to, okay, you can see this is about the origin. I want people to make research about the origin on their own. So when you make research about the origin, don't copy, don't take any screenshots. Be good students. When you make research about the origin, you make research about the Archaebacteria, find out what they are, the divisions of Archaebacteria, then make research about the Eubacteria, what they are, and the divisions of Eubacteria. Then after, this is just a summary. So for me, I don't need your summary. But after you talking about the two, you give me the differences. Yes, you give me the differences. Give me the differences. Yeah, you give me the differences between you and Archie. Then, these other things, I want to look at them at last because they deal with application, more of application. The economic importance of bacteria in an ecosystem on nature. Even these ones, when you read them, they are just very cheap. Then the other thing, that's about economic importance. Then we have commercial applications. Importance is the same as economic importance. In science, economic doesn't mean money. These ones are strictly and strictly commercial applications. And I don't even want to handle this for you. These ones are very cheap. The content is done. Then they are talking of why bacteria are abundant in nature. Yeah. And then that will, I think, be the end. You can say I told you that here it's where we shall have the next lesson. Now, we are going to make a simple deal. These things are not biology. I don't need to explain. You read and understand. But you do this. You take a screenshot of that, of strictly the importance of bacteria in an ecosystem or nature. You take that. I don't want to over-explain them because they have terms which we may need in other topics. But you can take a screenshot. Hope you're done. And then someone should share these after in the group of I don't know if you take this screenshot, it will be clear. You can let me know. When you take it, is it clear? Or we need to separate it into two? Okay, let's separate it. Let's non-test. Okay, you take that. I think that is very clear. People disappeared. Hey. For fear content. Then you also take that. You just read through those things. The curriculum just wanted you to know them. They are not the way you're seeing them here in textbooks. Most of them were obtained from the internet. Yeah? But the textbook information, I've explained it very well. Then also take that screenshot. So the next lesson shall strictly be on eukaryotic cells. Now, just because for us we just have 1 week of lessons, but this is what we usually do at Science Scholars Academy. I always, the work I give to you as research, we always create groups after, but we usually do this with holiday coaching and homeschooling. So for the premium students, we always create groups, because when I do it for these free classes, we have little time and people are not always serious. So when I give you groups, you present the work. We get online, you present, you share a screen. I hope you share a screen, you present with your groups. The way you divided the tasks, you start talking. As people are listening, as I correct your work, and as I critique, as I comment, and so on. So we cover it that way. So just know we complete the the syllabus together. Yeah, both when I come as the tutor, the lesson tutor centered, then that is student centered. Is it okay? Now, let me put everything here. The assignment. Yeah. So you people, thank you for listening. I don't know if people have gotten content. So let me end the class before people really completely burn out in a lesson that is supposed to prevent burning out of school. So the assignment is easy. I want people to tell me about the origin of bacteria and eukaryotic. Bacteria and eukaryotic. Then 2, you're supposed to tell me about you're supposed to define bacteria and explain its classes. Whether you call them types, whether you call them forms, I don't care. Then after that, you go to eukaryotic. So these ones are now the new divisions of the kingdom. The good thing I've told you the old divisions of kingdom Monera. Now you will really connect everything for yourself and things will come out. Yeah. So you're supposed to define the eukaryotic and also explain its classes, types of forms. Its classes, types of forms. Hope people saw that as part of the syllabus book. Then 4, you're supposed to compare. You saw my table, so I also expect a table from you. So in the notes I'm going to provide, I think I'm going to find ways of detaching. I'm going to find ways of detaching my table. So compare those two bacteria and bacteria. So I'm going to allow people to take some screenshots and then post them in the group. Yeah. So before I completely sign out or finally sign out, let me give you a chance to take some screenshots from the beginning. Say that you're not left out. Okay. Yes, you take. That is the first one. These pages are very dirty. Someone should leave my screen. But I'm going to find ways of extracting all this and sharing. take that. Take that. Okay. You take that. You take that. Okay. Oh, people have copied something. People have copied something. Don't want you to copy. Okay, you took these other things. Okay. Then to end with, you're supposed to read those things I've ended with. Those ones, they don't have content. Why would I explain importance? So, you're supposed to read importance. Supposed to read the commercial. I think it was commercial applications. You took the screenshots, eh? The good thing we're uploading this on our YouTube channel, so everyone will also be in position to take the screenshots. Commercial applications of prokaryotes. And they are so important. Never joke with application in your curriculum. It is so important than even the content. So you take it serious. That's why I'm asking you to read. Even put please. Because they can decide to leave whatever we have covered above and bring application. But for application to make sense, at least you should have the content we've said. So, why the bacterial abundant? I think that's the last screenshot we took. Okay. Now, after that, I think I should tell you our next stop. I don't know, maybe presentations we shall prepare on weekend and people present. I don't know. But our next stop about the research, I would want people to present. Presentation. What do we present? Told you the research. And then our next stop should be on the eukaryotes. Yeah. Okay. Now, let me sign out and you also take the screenshots of the workbooks. So allow me sign out as Dr. Paul. Or dude, students just call me doctor. And I like it. A member, a tutor at Science Scholars Academy, Uganda. Abbreviated as SSA. And for today, my personal number is just here. Yeah. So I think now you can also take some screenshots from the workbook. Okay. So if at all we don't manage to, do what? To present, those who are going to be under homeschooling with us, those who are going to be studying from home and the day scholars, be ready, you'll be the ones to present, because I believe you're also part of this. Because I will not repeat this, I'll just continue from wherever we shall have stopped with the students we are preparing for the term. So you take that one, that screenshot. Was the first one showing the divisions and everything. Yeah. And then lastly, it is this. So in whatever you're showing, this should appear. Because now this is the most important screenshot. It shows the assignment, it shows the reading, the next stop, and everything. Very important. Yeah, hope that is done. So maybe the other assignment, the basic one that I forgot, before you even do the assignment, just like in math, supposed to read today's content. Supposed to read today's content twice. If you read it once, you're not serious. Supposed to read it twice. Actually, the content has not been too much. Supposed to read it thrice. Yeah. You can complete the second and third time from school. You can read it once for now. And at school, you also read it for the second and third. But please do it thrice. Otherwise, thank you. Thank you for listening. Thank you for attending. Let me see if someone has thanked me for the lesson. I give them free classes for the whole senior 5 and 6. Let me try to read through the chat. Yeah, phycocyanin, phycoethrin, and the bacterial chlorophyll. Hey, Dark Angel is praying for me. The same way I usually want it. Thank you so much. So that's how we shall be moving. Every topic we touch, we shall be touching all content. And trust me, with the Notes of Science Scholars Academy, you don't need to read the textbook. No one should ever lie to you. I read all textbooks. I got all information from all textbooks. That's why the notes are worded. So you don't need a textbook. Those people who tell you need textbooks. Yes, you need them to get an A because you get some academic aura from them. You get. Yes, I'm seeing the appreciations. I don't know if I should give you all free classes. Yeah, I'm going to give you the recording. I think you'll find it tomorrow. Okay. Okay, people, thank you for attending. We shall meet when we next meet. About the lessons, I think all of us have taken screenshots and they are going to be posted in the Biology Scholars community. Maybe you ask for that. Then about the next lesson, you can check the timetable. Otherwise, yeah, we'll meet again, inshallah. Yep.